So that was a very interesting talk, and we'll have much more interesting talk again about along the same lines. I am happy to introduce Professor Rui Kuang. He's an associate professor in the Department of Computer Science and currently the director of graduate studies for the computer science graduate program. He works in bioinformatics. He got his PhD from Columbia University. I would rather read from here because I don't understand half the terms. So his current projects center around biomarker identification for cancer and disease genome analysis. And he's going to talk about this next.
Okay.
Please welcome Rui Kuang.
Okay. All right. Hello, everyone. So thanks for the introduction. Today I want to talk about some of the work in our lab and also along with some of the background about using computational methods for cancer genomics. And my focus is a lot on network-based methods and pretty much on using machine learning and some graph theory algorithms to look at the genomic data. So my talk will start from genomics and cancer, and then I will try to get into the algorithm part. It's a little broad, okay? So, but sometimes some of the things might not be all introduced at the beginning. So feel free to stop me if any term or anything is new to you. I'll be happy to explain. Okay. So, I actually started to work on cancer genomics about I think 10 years, 11 years ago, and I thought that was a very fascinating area where that's a huge amount of data, or a lot of them are high dimensional, and none of them are easy to understand. You have to put them together to understand a very complex disease. And in genomics and definitely cancer is the area where with the most diverse set of data and diverse set of information people try to put together. So a lot of new technology was actually, a lot of them were actually developed just for cancer genomics because this is so important a disease. Okay, so that's where the abundance of data people try to analyze and try to improve cancer treatment. So briefly, I actually want to start with some background about cancer genomics. So thanks to Chad, he probably already covered a lot about genetics and then hopefully some of the genomic technologies, but if not, I will talk about it a little bit. And then I will talk about using network-based methods for genomic data analysis. It will actually cover a broad range of machine learning methods and how they can be done in a network context. Okay, and then I will actually talk about some of the graph-based algorithms and then for association analysis. And I think Chad talked about how to link those mutations to disease, right? So in 23andMe, well, they don't have it anymore, right? So they report this disease gene or disease mutation associations. And then these associations, most of them are not validated. Okay, so that's why FDA actually shut this service down because they don't want people to panic on something that is not confirmed. All right, so and these associations, a lot of them are discovered from the data. Okay, so actually I want to talk about how these kind of associations can be analyzed in the network context. Okay, so yeah, these two category topics are a little bit specific, but we will also try to talk about the broader information about the methods and the data as well. Okay, so and at the end actually is also my ongoing research projects. I want to talk about how we apply in some of these multi-relational learning methods, actually using some of the tensor-based models. Okay, so all right, so I just start with some background. Cancer definitely is cancers are largest category of the disease. Every year, a lot of patients, and the cancer actually they are driven by this DNA mutations. Okay, so cancers are genetic disease, right? So it means this has to be something wrong with the DNA. So where people is widely believed, so the cancer is developed along this line where by accumulation of the mutations. Okay, so so in some of the environments, for example, some toxical environment, and the cell is in some toxic environment, or there are some other reasons. So the mutation rate is much higher than random. Okay, so then in some of the particular tissue or some cells, the accumulation of the mutations is much, much faster. Okay, and then because there are a lot of mutations, and these mutations, you start to acquire some of the mutations that could trigger cancer. And as you accumulate these mutations at some point, and then it turns on these biological pathways or processes, and then the cell acquires some of the abilities, such as proliferation, and then they actually grow into this, they become malignant cells, right? So one of the important characteristics is proliferation, where the cells never die, okay? So they will keep multiplying, and then eventually they become a big tumor. So that is the process where how the cancers are developed. So just to give you some concrete example, what might happen when a lot of mutations are introduced in the DNA. Okay, so on the left there, just a figure just to introduce when I say mutation, what it really means, right? So you could have some modification to the DNA sequence in human genome. So those could be insertions. So we're, in this example, A is inserted at the middle of this chunk of this DNA fragment. And then you could have a substitution, so say a C is replaced by A, or you could have some deletions, so C or one of the nucleotide is deleted from the DNA. So, and why this is such a small change would be problematic. Okay, so the picture on the right is actually a very typical case in cancer where an important gene called the TP53 is mutated. Okay, so TP53 is a tumor suppressor gene. Okay, so this gene actually turns on the cell death process. Okay, so this gene has to be active so that the cell can go into the right cycle, and then eventually the cell will die. Okay, so if something goes wrong with the TP53, you turn off the cell death process, right? So then the cell will stay there forever, right? It will start to multiply itself. So, and so on the right, what I'm showing you actually is the green, this tertiary structure is the TP53 structure. And so there are some of these yellow regions you see, and these are actually DNA binding regions. Okay, so genes are coded on the genome, right? And then each gene is translated into proteins. Okay, so those proteins are the power horse. Okay, so they actually bind to each other. They drive the functions and the biological processes, right? So if something wrong to the DNA and it translated back to the protein, and then the protein is doing something wrong to the whole cellular system, and then something goes wrong. Okay, so this is the case that in a lot of cancers there are mutations happens to TP53, and people actually observed in a lot of cases the mutations actually happens on the binding surface. So these are these. Let me see if I can get the mouse. Erase, I think it's. All right, I don't have the pointer. Okay, but. So the red molecule on the right is actually the DNA. TP53 is the DNA binding protein, which is transcription factor, triggers the transcription of some other genes. So if mutations happens on this, in these yellow regions, there are 11 amino acid residues in these regions. And if one of them goes wrong, it will reduce the binding efficacy of TP53 to DNA. Okay, so then which means the TP53 doesn't function anymore. Okay, and then in this case, and this definitely will become a cancer driver, and where the cell starts to acquire the ability of proliferation after TP53 is mutated. Okay, so that's only one example. Okay, so in some of the cancers, TP53 is frequently mutated, right? And which to the doctors is easy because I always check. I know that 95% of patients would have a mutation in TP53. So then targeting on that, I can design some treatment. Okay, but in some of the other kind of cancers, it's not so obvious. Okay, so actually they are the mutations could happen in a lot of different kind of genes. They all can be different cancer drivers. Okay, so and TP53 is the most well studied, one of the most well studied case, and in most of the cases, the mutations and the drivers are unknown. Okay, so actually this picture actually shows you a wide range of the genes that could be associated with cancer. Okay, so those ones that could trigger cancer, be cancer drivers, they are called oncogenes. Okay, so and because they are these mutations in these genes could drive cancer. And then there are tumor suppressor genes, and then like TP53 is one of the examples. And then there are other genes in different functional categories. So for example, if one of the gene which does the DNA repair goes wrong, so then which means when the DNA is copied, there's no correction. Okay, and then you start to get a lot of mutations in the DNA. All right, and then that's a way of accumulating a lot of mutations, and then you develop cancer. So, and then there are other carcinogen and cell cycle genes, cell cycle checkpoint genes. So it's actually a long list. So people don't know how long the list is. Okay, so I think at the time I started to look into cancer, and people think there are like 200 genes, and now there are gene lists as long as 3,000 genes published by some research institute. Okay, and then eventually people agree, oh, it's meaningless to publish cancer gene list because there are just so many of them. Okay, so how many genes do we have in human genome? 3 billion base pairs. Okay, so human genome is 3 billion base pairs, so 23 pairs of chromosomes, right? And then on the genome, and there are these chunks, right? Each of these chunks, they code for genes, right? And actually, human genome codes for 22,000 genes, around 22,000 genes. And we actually don't know that until almost 2005, about 10 years ago. And then we get the exact number. We have around 22,000 genes on human genome. So, and potentially, all of them can be contributing to cancer. Okay, so there are some of the ones in these well-known categories. But again, when you start to look at the combinations, okay, so it becomes very, very long. So, and then this becomes very difficult, where the mutations in each of the patient is different, and they could be driven by many different genes, but you have so many candidates. Okay, so identifying them becomes really difficult. Okay, so actually, the good news is that in the past about 20 years, and especially in the last decade, there are a lot of very exciting genome-wide genomic technologies have been developed. So the goal is to apply these technologies to look for these kind of mutations in the DNA. Okay, so that if you know all, you can reveal all the mutations, and if you can identify which ones are contributing to tumor, and then you can start to design those targeted therapy for a particular patient. Okay, so earlier days, people use a technology called a microarray. Okay, and microarray is the first kind of large-scale technology. Before people have microarray, biologist or doctor can only check one gene at a time. Okay, so and we have 22,000 genes, so it's very inefficient. So usually, when you look at the 10 genes or even 100 genes, then it's a very large-scale study. Okay, so microarray was the first one that actually was designed to measure all the genes simultaneously. Okay, so and then later the microarray is designed based on the idea where there are complementary sequences you can bind to the DNA. So you design some array where you have a signature for each gene you want to measure. So then when you hybridize the cell extract on the array, and then the DNAs start to bind to the signatures. Okay, then you can count how many you have. Okay, so this is array-based technology. And then in the past 10 years, another more exciting technology is the sequencing technology that was actually developed. And this technology is better than array-based technology because you don't have to know the signature of the genes. Okay, and then you just collect the DNA, and then you just read them. Okay, so you read a lot of small fragments from the DNA, and then you can put them back together as a whole genome. So then you actually get all the mutations or all the other kinds of aberrations in the DNA. Okay, so sequencing technology actually is much more exciting, especially for cancer genomics. So the reason is because you really don't know what the mutation is. Okay, and then array-based one, you have to design the probe that are complementary to what you want to design. But if you don't know what you are detecting, then the array technology is not as useful. Okay, versus the sequencing technology is de novo, which means I don't care what's in this genome, right? I just keep reading it. I read it many, many times, so I can put them all back together. Then I can read all kinds of aberrations. Okay, I think Chad talked about he sequenced his family sequenced their genome, right? And then they're looking for some of these mutations. And even if they are not in the database, and then using sequencing, they can reveal it, okay? So, and with these technologies, and people actually organize the very large-scale genomic studies for cancer. So what they do is they apply these genome-wide technologies to look at a big patient cohort. Okay, so for example, there's one national project called the Cancer Genome Atlas. Right, they actually collect the DNAs and RNAs from. I think right now they have. I don't want to be wrong, but I think the data already in the database is more than 23,000 patients. Right, so but they are keep collecting more. And eventually, they want to get it to 50,000 in a couple in the next few years. Okay, so then the goal is that once we collect all these data for the 50,000 patients, so we can compare their genetic variants together with some interesting outcomes. Right. So for example, I have breast cancer. I have 1,500 patients. 100 of them have a good reaction to a treatment. 500 have a bad reaction. Right. So why? So now I look at the genetic variants I identify from this 1,500 patients, and then try to figure out which one can predict the response, right? So now if you have a new patient, you check the genetic variants, and you find out, oh, this patient has genetic variants matches with the one with good response, then this patient should be treated by this particular disease. Okay, so that's where it starts to give you a lot of power. So, all right. So just to want to talk a little bit more about the genomic technology. So do you think reading DNA is very easy? All right. It turns out to be extremely difficult. Okay. So, and all of you know about the Human Genome Project? It's already in past tense. So it was a big thing when I was a PhD student. Okay. So, and that was the first project to sequence one human genome. Okay, so now if you want to sequence one human genome, it takes a few days, $1,000. And back at that time, in the end of 90s and beginning of this century, it cost $3 billion. Okay, so it's $3 billion at that time. So and the main difficulty is that because the DNA sequence is so small, and you have to design some sophisticated technology to read the bases, right? And then because they're so small, so you have to collect a lot of information and put them all together. Okay, so but it is so long. When the reads, and once you get past a few hundred base pairs or a few thousand base pairs, you lose the signal. Okay, so and the Human Genome Project was the first one trying to have a collective effort from thousands of, I think there's almost a few hundred participating labs, I think, from many countries. And then each of them is assigned one fragment. Okay, so they're just responsible for sequencing part of it. And then at the end, they put all of them back together, and then they assembled the first sequence the human genome. Okay, so and the reason why it was so expensive is because the device to read the DNA can only read can sequence one sequence at a time. Okay, so you get one DNA fragment, you start to sequence it. Okay, so then which means if you have 10 machines, you can only read the 10 sequences at the same time. But we have 3 billion base pairs to read. Okay, so so around 2005, 2007, and where people call this next generation DNA sequencing technology. So the idea is that now, rather than reading one fragment at a time, we actually read millions of fragments simultaneously with the machine. Okay, so with that development of the machine, so really the sequencing speed is 1 million times faster. Okay, so then that's why now we can actually sequence human genome with just $1,000. So I remember at the time of 2012, people still don't believe you can sequence the human genome with $1,000. Right? And now it's true. Okay, it's only after 5 years, right? So, and why this is so important to cancer genomics? Okay, so that is because again, if you want to know all these genetic variants, right, all the ones in this patient, you have to read the whole cancer genome, right? The cancer genome is different from the normal human genome, right? So, so I think Chad talked about the single nucleotide polymorphism, right? And these are the differences between the DNA in, say, healthy people, right? So usually, so say if two people in this room, so how many base pairs difference we are expecting between their genome? So human genome is 3 billion base pairs, right? If you take two healthy people and compare their DNA, so they won't be the same, right? So how much difference they have? 23%?
No, 0.3.
0.3. Okay, I'm trying to do a calculation. It sounds still a little bit too big to me. So usually we are talking about the millions of base pairs to probably at the most 10 million. So that's the scale we're expecting. So divided by a billion, so 10 to the 9, 10 to the 7. Yeah, maybe you're right. It's getting is the right ballpark. Yeah. So that's the amount of differences that you are expecting. Okay. So and if that's a small amount, the good news is that we do not really, if you want to know a new genome, right, you do not really have to sequence the whole genome because most of them are the same, right? So I can take an even very low coverage, and then I just compare this one with some known ones. I can impute a lot of information, okay? So, but for the tumor, and this is very different, where in tumor, because there are a lot of mutations already introduced, so this cancer genome is very different. Okay, so and then you would have to sequence the whole cancer genome to know why, how different it is, right? Some of the cancer genomes can look very, very different. It's not even 23 pairs of chromosomes, right? And then they are shuffled around in some very strange way. So that's DNA sequencing becomes very, very important for cancer genomics. And in a lot of sense, a lot of development in cancer genomics was actually driven by this sequencing technology, right? Because now we can get the whole cancer genome. Now we can talk about looking into the variants. Okay, so, and in this picture, I just want to give you a sense about how DNA is sequenced and how they can identify the variants. Okay, so did Chad talk about this? No? Okay, a little bit. Okay, so I will still cover this slide. Okay, so basically this is an example. If you look at the top, and this is genomic DNA from a tumor sample. All right, so and which is one chromosome. So this chromosome is different. Okay, not surprising. And you can see that I think on the shoulder arm on the left side, there is an inversion. Inversion means one part of the DNA is inverted, okay? And then the next one, on the short arm, there's a deletion, okay? So then this genome actually lost a part of it. And then on the long arm, there are some places called a copy number gain. It's one region is duplicated multiple times, okay? And then there are SNP, so it's a single nucleotide polymorphism. There is one of the particular nucleotide is mutated to something else. And then there are some translocation. And that's actually a combination of two segments from two different chromosomes in a new chromosome. Okay, so then these two chromosomes are different colors. So one of them is green, and the other one is pink. Okay, so now in sequencing, what we do is that we first we break the DNA, the chromosome, into a lot of fragments. All right, so then you get a lot of these shared genomic DNA fragments. So and the colors actually tells you about their origin. Some of them from the translocated fragment from another genome, some of them from this genome. Okay, and then for these DNA fragments, so now we can use this sequencing technology to read it. Okay, so what's interesting about the technology is that it's not reading the whole fragment. It's actually reading from the two ends. Okay, so you have the 5' end, 3' end. And then the technology will start to read from the two ends. And then they can go ahead and read about, so say 250 base pairs on the 3' and 250 bases on the 5'. And they get these pairs of the reads. Okay? So now you can see some of these pairs of the reads are actually already interesting. So for example, I have a read that the part of is I think it's blue. And then the high level is pink, right? So you know where they are from? If I go back to the original cancer genome? So yeah, it's at the translocation, because partly on the original, the long chromosome, and the other one is from the other chromosome. Okay? And then you have some even more interesting ones. There's on the fourth column, you will see the first one is actually exactly the left read is exactly going across the boundary on the translocation. Okay? And then, and now you have these base pairs, these read pairs. Now you can use a computer algorithm to align these read pairs back to a reference genome. Okay, reference genome is a healthy genome. Okay, so now you read the align the read back, and now you will see this alignment of this short read compared with the healthy genome tells you a lot. Okay, starting from the left, you see those reads that in this in the red rectangles. So these are the reads that actually from the inversion. Okay, so once detecting, you see these reads don't look right because they go in the other direction. Okay, so then you will see that deletion and is also in some of the reads because these reads also skipped one of the positions in the healthy genome. Okay, so then you know, oh, in the cancer genome, this position is lost. Okay, and then you move ahead, you have the single nucleotide polymorphism, and you will see some of the read actually tells you that this is a G. Okay, so then really in the reference genome, this should be a T. So then there's a substitution happens here. And then you go all the way to the boundary of the translocation. So you start to see the cases, one read is on one chromosome, another read is on another chromosome. Okay, so then by putting all this together, and actually you can come up with a list. Okay, so say, oh, these are the mutations we actually observed in this cancer genome, and these are the possibilities where the drivers of the cancers we're looking for. Okay, so that's how you can identify these variants. So any question about it, how this happens? Oh, it's clear? Okay, so this is kind of like an ideal case where I assume mapping the reads is easy, okay, but in reality, so these abbreviations can be very complicated. So actually a lot of very sophisticated algorithms were actually developed just to identify each of these kind of variants. Okay, so that's actually one big area in cancer genomics is how to make this variant calls. All right. So, so now with this sequencing technologies, there is a huge development in cancer genomics. So basically, now we have these powerful tools. We can check for all kinds of molecular information from tumor cells. And then hopefully with a patient cohort, we can identify these drivers or targets for treatment. So primarily starting from the DNA, and by sequencing or array-based technology, we can collect those DNA aberrations, mutations. So for example, the point mutations, the translocations we have seen, and then we can also report those copy number changes, right? So duplications of the fragments. And then there are also something called epigenomic alterations. This is actually has to do with the DNA structure. Okay, so it turns out that it's not only the information in the genome plays a role, and how the genome fold take the 3D structure, and also actually plays a role determining which genes are actually active in the tumor cells. Okay, so that's people also collect this epigenomic alterations. So collectively, and these are the information on the DNA. Then eventually, after these DNA changes are introduced, say they are going to change the cellular system. Okay, so again, what is observed at the cellular level is actually the molecular activities are different. Okay, so which means in the normal case, the cells will maintain a mechanism, make different number of copies of the proteins, right? So if you are getting the proper amount of good proteins, normal proteins, and then the cell function normally, right? But then once you have these changes, some of the proteins don't work anymore. Okay, so if they don't work, and it will actually change this whole cellular system, and then the activities will become different. So, and people actually collect the information about transcriptons, and these are the RNAs where the genes are transcribed into RNAs, then into proteins. Okay, so people measure the RNAs, so they actually give you a lot of information about the protein levels. And then people also directly measure protein levels in some of the cases, but usually it's still very difficult. Okay, measuring protein activity is very, very difficult. Okay, so people usually look at the RNA activities, so which is called the transcriptome. And hopefully that these transcriptons can tell you about the cancer cells, okay, at the cellular level. Okay, so it's a system. So I think one key message here is that mostly you probably hear about these variants, right? So that's because the change is introduced into the DNA, but those variants do not play a direct role. Okay, so when I say that these variants do not play a direct role, it's because they don't do anything. Okay, they just give the information. All right, and then this information they are translated, and then that's where they start to play a role. They are translated into the proteins. As I showed to you, the TP53 is the one that actually will start to have some problem. So that's in cancer genomics. Looking at the genetics is one big area, and another very big area is actually the systems biology approach. Okay, which means I don't know what is in the DNA. Okay, but I'm just looking at the molecular activities because there are a lot of changes to the DNA, so the activities look different. Okay, so now I just by looking at the activities, I will actually also be able to tell you. So which means, so for example, I'm just again looking at the 1,500 breast cancer patients, right? And then I can look at the molecular activities for each of the patients, and then I'm trying to identify which molecular activities actually leads to a drug response. Okay, so that's is looking at this at actually the systems biology levels. So actually, what is surprising is that in most of the studies, people have already observed that the information in the systems biology level is much more predictive than the genetic variants. Okay, so in other words, if you want to predict drug response, predict some outcomes, metastasis, looking at the molecular activity is actually more informative than looking at what's wrong in the DNA. Okay, but biologists like to look at what's wrong in the DNA because if you know what's wrong in the DNA, you directly know which gene is wrong, and you can see whether there's some structural change. Okay, but at the system level, it's very difficult to explain because there are 22,000 genes, right? And why their activities are changed. Okay, so that's a big area in computational biology. People try to look into if you modify some of the genes and how the molecular activities will change. Okay, so that's another big thing that people work on. And actually, a lot of these discoveries or useful tools were developed based on this transcriptome analysis. Okay, so right. So now I want to actually get to a little bit about some work in my lab. Okay, so again, this is like my first project on cancer genomics. And so I was very interested in looking into a high-dimensional data set for breast cancer metastasis. Okay, so at that time, that was almost kind of one of the largest data set. Okay, so it's I think around 100 patients. Okay, so and then one of the first thing actually I did is that. Because 70 patients is a small cohort, right? And I have 22,000 genes to study. Okay, so I think Chad mentioned about this multiple testing problem, right? So we're in statistics, you don't have enough power from the data to make significant calls, which means most of the discoveries will be random. Okay, it's not because they are really a driver. So what I did is that I was trying to pull out more data, right? So I say, oh, I have just a similar data set from another hospital, and then I have a few others. So I want to put all of them together. So presumably in statistics, if you increase the sample size, and your discovery is much more reliable. Okay, So, but what is very, very disappointing is that I actually tried to look at, my lab actually looked into the important predictive markers from multiple data sets. And when I put them together, I hope they will tell me one coherent story, but in reality, they are completely inconsistent. So for example, if I look at the top 100 most relevant biomarkers, like from, so say, 3 data sets, I compare these 300 genes from the 3 data sets, the OLAP is 0. Okay, so, and that becomes really very disturbing, right? So you hope you can find some things that is predictive, right? And then what you learned from one data set is completely useless in any other data set. So and then that's a big problem. So, and actually the data set that we analyzed, the one of them, is a data set, I think it's around 108 patient samples. They discovered 70 genes as the biomarker. These 70 genes are actually, it's called the mama print. If you do go to the wiki page, and you will see that mama print was actually the first approved, I think it's a clinical signature. So which means the doctors can use it in the clinics approved by FDA. Okay, it was discovered around 2002, and it was approved in 2007. So, and a lot of reasons that why there are a lot of delay is because a lot of people don't believe the 70 genes is just the reason I mentioned, because it's not reproducible. Okay, you look at another patient cohort, none of them are in the 70, right? And why this should be a signature. Okay, and I actually on the figure, it's some of our analysis. So we use 3 different kind of ways to identify I think it's from 2 different data sets. And we try to see if we find the same biomarkers from 2 data sets. So you can see at the top 100, the overlap is 0. Okay, so it's only until about 180 you start to see some of the overlaps. So 2 or 3 genes that actually are in common. Okay, so so that actually directly reveals these challenges in the biomarker identification, where we have a lot of genes, we don't have many patient samples, and then actually. One of the learnings in my early experiments is that it's wrong to look at each individual gene, okay? Because these genes actually they work together, right? So they function together as one module, and then there are interactions among each other. So if you are looking at individual ones, the information is incomplete. And the high-dimensional data, these genomic technologies are usually highly noisy. Okay, so then when you have different noise on individual genes, they are not robust markers. Okay, so and so that using these gene modularities is actually will be very important. And then and again, just because of these high dimensionalities, and with the noise and all the biases, so you would be expecting you have a very high false positives and high rate of false positives and false negatives. So if they are all wrong, and when you compare them all together, so it's not very surprising that they are different. Okay, so and then also there are patient heterogeneities, because a lot of variants, they are actually relevant to something else, right? So maybe gender, or maybe some hidden clinical variables which you don't know. Okay, so then these ones actually they play a role and has nothing to do with cancer response. But because you have a small patient cohort, and then you start to see, or you think there's a relevance, and it's actually something else. Okay, so got a question?
Yeah, your point about looking at an individual gene isn't really reliable. You have to look at the combination. So how far does that stretch? Like, we always hear about, you know, if you have this gene, you're going to have blonde hair. Like, is that still reliable to say I can look at this gene and figure out what traits are going to be expressed, or do I have to really look at combinations in that case too?
Okay. Yeah, so that's a good point, right? So when I say you have to look at the multiple of them and what it really implies. So usually people call the diseases or phenotypes that are relevant to more than one locus. They call them complex disease or complex phenotype, right? And then there are a lot of simple ones, which means it's one mutation, it's one gene. You already know it's going to lead to this phenotype. But there are many, many more cases that actually are combinations. So cancer is one of them. Yeah, it's not a one. In most of the cases, it's not one mutation. So a lot of them actually play a role. Yeah. So in here, when I say that individual genes are not reliable and informative, and it's actually also because sometimes it's a collective activity is giving you the signal rather than one gene is active or not. So you might have a pathway is maintaining some biological function, and if you look at the one gene, it might be normal. But if you look at the whole module, it's not functioning. So without looking at all the genes, you don't know that actually the whole module is not working, rather than just the one gene. Yeah. And a lot of these factors are put together, and making the statistical analysis actually is very, very challenging. Okay, so I'll just get into this point where, again, this is our first project, and then we actually did something really simple. We actually applied a category of graph-based semi-supervised machine learning algorithm. So the idea is that when you are trying to looking for the biomarkers based on some patient cohort, okay, and you are not just looking at the one gene at a time. So we actually build a graph, and the graph actually contains the information about the group of the genes. Okay, so when you try to identify which gene is important, you are looking at all the genes that are relevant to this gene, and then you summarize this information with an algorithm. Okay, so we actually applied two kinds of algorithms. So one of them is a bipartite graph approach. So where your original data is a matrix. So so say you have group 1 and group 2 patient samples. So group 1 are the patients that with, so say, a positive response. And then group 2 will be those patients with negative response. Okay, so now then you have the gene activities. 1 means the gene is highly active, right? So highly expressed. And 0 means it's not expressed or very low expression. So now I build a bipartite graph where on the left I have the nodes as the patients. On the right I have the nodes as the genes. And then the connections are the weighted edges by the gene expressions. Okay, so once I have this representation, so I can apply graph-based machine learning algorithms. So these algorithms, they are actually looking for those things called bicluster, which is a subgroup of patients and a subgroup of genes which are highly associated with each other. And then the algorithm iteratively summarizing this group of patients and a group of the genes, and at the end is going to accumulate this group-based information to rank all the genes to identify as the biomarkers. Okay? So, and then there's another way we can also build a graph just among the genes. And then when you rank the genes, so you look at the genes' neighbors, right? So if I have one gene and this gene is highly correlated with another neighborhood, right? And then the information will be smoothed within the neighborhood, okay? So then you have one big graph. There are many hidden neighborhoods, right? You just run one algorithm to summarize all the hidden neighborhood, and then you get the collected information. So the whole idea there was rather than looking at the one gene, look at the global information, looking at the context, okay? So, and actually we had just some quick results. And then after we apply this one, immediate observation is that now the results you actually observed from independent data set are highly consistent. Okay, so these are the same data set we actually analyzed. So the x-axis is the number of genes you actually selected from a data set, and the y-axis is how many of them are the same. Okay, so you can see the top curve is this graph, bipartite graph-based algorithm. So in the top 100 genes, 40 of the genes are actually the same. Okay, and that's that was actually quite encouraging because immediately we saw there's a value rather than looking at the 24 independent variables. I'm looking at their relation, right, and ranking them. And now these data sets start to look somehow similar to each other. Okay, so and then along that line, actually, my lab actually worked on a collection of these network-based methods. We try to analyze this gene expression data in this network context, and then we'll hope that within the context, we can build better predictive models. We can identify more variants that are actually relevant to the cancer treatment. Okay, so and this is a figure actually from a review article we wrote last year. That review article was on a bigger context where we are talking about both using network to analyze the genomic data and also doing some association analysis, which is the second topic, and to predict which drug actually can target on which gene in the network. Okay, so and then we actually also reviewed some of the category of the algorithms. I think after 2007, 2008, before that, people didn't realize network is important. Okay, so then after that, network analysis becomes a standard analysis in cancer genomics. So now you read every cancer genetics paper, there has to be a section talk about network analysis. Okay, so somehow they have to use the network to analyze the data. Otherwise, it's incomplete. Okay, so that is actually becoming very popular. So, and our paper actually kind of summarizes 3 different ways how to put the network together with the genomic data so that your analysis is more robust, and then you actually identify more consistent results across the data sets. Okay, so that's one of the interesting trend. I think still people believe that this is definitely a necessary step, right, to look at the data. So I just want to skip a little bit, just to jump to one of the model we developed. So in one of the projects, we worked on this machine learning-based model for survival analysis. So and this is some statistical model. We're trying to predict some particular risk. Okay, so it's giving you have some patient data, and then you will actually predict for some of the patients at a particular time what is the risk of an event, right? So for example, a death or a recurrence of a tumor. Okay, so rather than using the standard model, which is a Cox regression model, we actually introduced this network information and as a constraint in the original model. Okay, so the constraint we're using is a Laplacian constraint. Okay, so we actually also started to use it around 2008, and then now it's also become a standard method. So when people want to integrate the network, they use Laplacian constraint. Okay, so and also a lot of statisticians did follow up. They actually analyzed why you can use a Laplacian constraint. So our motivation was completely empirical because I want to use network and put it in the model. But then statisticians actually looked into why a Laplacian will play a role. They actually explained it in a statistical perspective. Okay, so and then we actually introduced this additional constraint in the model, and now rather than just looking at the individual gene expression patterns, so we are looking at the network information as a context, and then we try to identify the important genes. So we have some encouraging results. For example, the marker genes are more reproducible across 5 different data sets. Okay, but this is identical to what we discovered before, so it's not surprising. And then we also identified some of these sub-networks, which suggest that the neighborhood actually is playing a role. So these are exactly those hidden modules I actually mentioned about. So it's not known. So after you apply the algorithm, you can actually find these are the modules that actually should play a role. So actually, I just want to jump to a very interesting discovery in our study. Is this particular gene called FBN1. So FBN1 is actual cellular matrix receptors. So for those ones who don't know the molecular biology, actual cellular matrix is this molecular structure actually maintain how the cells interact with each other. Okay, so you have a lot of cells, and then the cells actually attached to each other through this actual cellular matrix. Okay, so FBN1 is one of these genes. Actually, it's a receptor, and it plays a role for this microenvironment, so how the cells are put together. Okay, so and what's interesting is that the data we analyzed is ovarian cancer, and nobody has reported anything about the FBN1 as an important gene. Okay, and our model actually consistently rank FBN1 at the top 3. Okay, it's either top 1 or top 2 or top 3. So and that was a very interesting observation, and then we looked into this particular gene to see why it's ranked so high. Okay, and then that's where we actually discovered that FBN1 is this one gene actually is in a big neighborhood. Okay, so which means FBN1 actually has a lot of neighbors, and these neighbors in this neighborhood actually play very important roles. Okay, and then when we do this network analysis through the modules, and they all point to the possibility that FBN1 should also play a very important role in some of the patients. So, and we actually looked into the data, okay, and my collaborator, he was at the Mayo Clinic and now in New Mexico. So he actually has another patient cohort, and then he actually did a validation of FBN1 with a protein staining approach, okay, on another patient cohort. And they actually observed exactly the same trend where FBN1 tells you about the survival. Okay, so the FBN1 higher ones actually has a higher survival probability, and it should be a real biomarker. And this observation is actually reproducible in another 4 data sets. Okay, so and then the question is that why the other methods don't report this gene as an important gene, right? So if you look at the gene expression in any of the data set, you use statistical methods without using the network, FBN1 is nothing. Okay, it's ranked at a few hundred. It's never at the top. So and my collaborator, and he has access to patient data, and he has a lot of knowledge about ovarian cancer treatment, and he told me that. This pattern is very important. It's very interesting. It's because FBN1 is not a biomarker until 18 months after the treatment. Okay. So and I think I don't have that full one to show you. So you can see all these plots. I actually start from the 20 months, and the FBN1 is actually only play a role after the outcome of the first treatment is known. Okay, so which means in all the patients, FBN1 doesn't come out as a significant biomarker, but after the first treatment in the 18 months, some of the patients had no response to the treatment. So these patients, they actually left the cohort because they died, right? And then in the remaining patients, these are the ones that actually have response in the first treatment. FBN1 starts to play a big role, because in this group of patients, FBN1 actually will be a biomarker, right? So that actually exactly explains that why the other methods will not identify FBN1, because it's only a marker for a period of time rather than the whole period of time. So you have to look at this subgroup of patients, then you will find the FBN1 was a significant marker. Okay, so this is just one example in the real study where we just show there's a value. Okay, so when you look at the network and then there really will be a value, you can find something that cannot be detected by standard methods. Okay, so that's just a question.
What does it mean for it to be a biomarker after 18 months but not before? Like, what's the difference?
Okay, the difference is the difference in the patient cohort. So typically in ovarian cancer, so these data sets, they all collect the gene expression data before the treatment, right? And then after they applied the treatment, and they start to do the follow-up of the patients, right? And then some of the patients will have bad outcome, which means they don't have a response to this treatment, right? It could be a chemo treatment. And then these patients will die. They leave the patient cohort. And then after 18 months, you have a smaller patient cohort. And for this smaller patient cohort, FBNI plays a big role, because these patients, really, the expression matters for them. It doesn't matter for the patients already died between the first treatment and the second one. Okay, so that's why I say it's only a biomarker for the second group of the patients. Okay, so and these kind of markers usually is very difficult to discover, because you don't know where to cut then. And then there are so many of them to check. Yeah, and then if you do that, you will get a lot of false positives as well, because some of the random ones will pop up as the important ones. But by using network is kind of, it's propagating the information in a reliable way, because there's a neighborhood information you are utilizing. Yeah. Okay, so that's some validation we have done. I think we currently get 10 to 15 minutes left. I can go for the next topic. But do you have any question about the first part? Okay. So now I want to talk about briefly talk about our second application using machine learning for identifying cancer-relevant disease associations. So this is a different problem where rather than utilizing the genomic data of the individual patients, we actually want to use a knowledge base. Okay. So when I say the knowledge base, in a lot of sense, I'm actually talking about some of the documented associations. Okay. So I think again, Chad showed you this table, right? So this genetic variants indicated this disease, right? And then you get a big list of those ones. And where these associations are from, it's from a database. Okay. So here I'll just show you an example. Again, this is from one of our early papers. So this graph actually shows you the relation among the genes and the cancers. So on the left, there are 7 different kinds of cancers in these rectangle nodes. And then on the right, you have 8 genes in these circles. So these genes, again, there's a network among the genes, tells you how the genes work together. And then among the cancers, you can also link those cancers that are more relevant to each other to build a graph. Okay? And then there are edges going between the genes and the cancers, and it tells you this is an association. So those solid lines between cancer and the genes are the ones actually they reported before May 2007. And then in the next 4 years, and then the dashed lines are the new ones that they added into the database. Okay, so what happens in the literature is that there's a research group. They read all the cancer genomics literature, right? So if there's a new paper, they read the paper and then see if there's a new genomic data set suggests that this gene, this variant, should be associated with this cancer. So they add this association into the database. Okay, so it actually is manually maintained. So because there are more and more studies about the cancer, and these associations actually grow really fast. You can see that it's almost doubled in 4 years. But now it's 2018. I think at the time I started to look at this database, there were about 2,000 associations. So now we are talking about 5,000 more. Yeah, I think it's probably already 7,000. Depends on which set of the disease you are looking at. So. And then this becomes an interesting question is that now this database is big enough, right? And when we don't have this database, your study is completely is not organized, which means I don't know which genes are important. I'm just going to do some whole genome profiling and then find out which ones are the important ones. Then I'm going to focus on the important ones to see which ones are should be further tested. Okay, so but now we have this large database. So for example, if at before 2007, if I utilize this solid lines, and then it will actually tells me that ovarian cancer should be associated with BRCA1, then there don't have to be 5 other studies. Actually, they all report BRCA1 is important because I predicted it's important. So then you just organize one focused study, and then you can show BRCA1 is important. Okay, so that's people call this gene prioritization problem, which means if you want to study a disease, and which are the genes, top genes you should look at. Okay, so if you can utilize this database to identify a reliable potential list, then it reduces the effort, right? So then you don't have to waste a lot of time and money to collect too much data. So that's one of the interesting work, actually, again, in my lab, is actually to look at this knowledge bases represented as a graph, and then try to see if I can do this kind of association prediction. Okay, so and actually in our review article, we reviewed several different categories of algorithms, graph-based algorithms. So basically, these algorithms are trying to utilize the structures in the graph so that they can make a prediction. So if you look at A, this is one example where this is actually, rather than using disease and the gene, I'm actually using the JOC and the genes. So on the left, this is a network of the JOC, and on the right is a network of the genes. So, and then there are green, so some of the associations, so you try to predict. So the way it works is that you can try to connect a JOC and a gene, right? And then you can actually calculate the shortest path going from one JOC to a gene. So if the shortest path is very short, that means there might be an immediate impact, right? So which means this JOC might be effective for treating this gene. Okay, so these are kind of simple methods using graph connectivity. And then our methods usually in my lab, we use the second category of methods, which is called link prediction methods. So we are not simply checking how far you are to nodes into networks. Okay, so and again, our idea is that you should look at the global connectivity between the networks. So if you look at the graph B, again, I'm using JOC rather than disease. So if you have the JOC network, you have the disease network, and then you have a lot of these green are the known associations, and then the red ones are the associations that you are trying to predict. Okay? And then you can utilize a category of machine learning algorithms. Usually they are called link prediction methods. So the idea is that when you are trying to predict a pair of association, you evaluate the global impact of adding this connection. Okay, so if you add this connection to the between the two networks, and do you actually reduce the path length between a lot of important pairs? Okay, so and we're using some of this random walk and label propagation based machine learning algorithm to summarize all the information. All right, so if you compare the methods in category A and category B is an enhanced version, right? So I'm not looking at the one pass, I'm looking at all of them. I summarize them in a meaningful way so that the prediction is more reliable. Okay, so then there is also a third category of algorithm. My lab don't work on those ones. The problem with the third category is that they actually do the semi-supervised learning, which means they have some so say some target genes, some of the genes are not target. You build a machine learning model to classify the remaining ones. Okay? But the main problem is that usually for one JOC, you just know 2 or 3 target genes, right? And then you are required to train a machine learning model with 2 or 3 positive samples. It's not reliable. Okay? So usually it's only applicable to widely used JOCs. Okay? So that's we use this link prediction methods. I just want to give you a quick sense about what really happens. So the philosophy of these algorithms are actually very simple. So basically, all these algorithms are actually exploring some subgraph patterns. Okay, and then they call this guilty by association. So there are 3 kinds of patterns that could be immediately interesting. So the first one is a case that where you have 2 similar diseases, and then both of them are actually associated with one gene. All right, and this is the cases because these 2 diseases, they might be relevant, right? And there are some molecular mechanisms shared between these 2 diseases. So then it's not surprising they might share the same disease gene. Okay, so the implication is that a similar disease might share the same disease gene. Okay, and then you can have the second case where so you have one disease which is associated with a particular pathway. So then you have 2 genes that actually both of them are in the same pathway. So then you know, well, because I know the disease is associated with gene 1, but gene 2 is playing a similar role in a function. So then gene 2 should also be associated, right? And then you could have the rectangles where similar disease, so one of them have a particular target gene, and then there's another gene is interacting with this gene, and then it's possible that this gene plays a role in another disease. Okay, so these are just kind of very simple graph patterns that people are looking for. But the problem is that there are too many of these patterns. Right? How can you summarize them in a meaningful way? So and we actually design an algorithm called a birandom walk basically this algorithm is counting this kind of patterns in the graph with a random walk. Okay, and there are some notations here. Basically, A is the bipartite graph, P is the disease network, and G is the gene network. And then you are doing a random walk multiplication on the left side and the right side. And you keep doing the multiplication. And each of these multiplication is actually counting how many triangles you have, how many of these rectangles you have. Okay? And then after all this random walk, you end up with a counting. If you compare one pair of gene and disease, how many triangles you create and how many rectangles you create, and you sum over them, right? And then there's a weight that the longer path or more distance relationships is downweighted by a decay factor. Okay, so that's kind of is an algorithm. It's actually extremely simple, and surprisingly, this is the mathematical framework, which I'll just yeah. So, but basically, it's surprising that the model is very simple, and the results are very good. Okay, so and it's because a lot of information really in the network. Okay, when you summarize the neighborhood, and you start to get a lot of very useful information, and the prediction performances in the benchmark data set is usually very good. So among the three categories of algorithms, so link prediction methods are still mostly best performing ones. Okay, so the best performing methods usually they would involve some step for this link prediction or global structural exploration. So in one of our studies, we actually look at the colorectal cancer, and then this is all the genes that actually were actually reported, documented in a colorectal cancer pathway. And the red ones are the additional ones, actually the additional associations our model actually predicted. Okay, and then we actually, what we did is that this is a work in 2012, and then we just go to the literature and see if there's a new papers about these genes, right? And most of them actually, a lot of them we actually found that recent papers actually report that they are relevant. Okay, so again, this is just to say that with this kind of methods, you can do more targeted study of the disease genes, right? And if you know which ones are the important ones, then your study can really focus on those ones. Okay, so and then just at the very end, my lab is still working on these problems, and our next step is to explore higher order relations. So when I say higher order relations, is from the network perspective, because network is pairwise, right? I have a pair of genes, and then they work together, and then there are cases we know that it's not only in the pairwise relations. So, for example, if you remember one of the first few slides, the genetic factors, including those mutations, copy number variations, epigenomic factors, all of them will play a role. Okay, and then you have to look at these combinations. So and to look at these multi-relations is immediately computationally challenging. And if you will see that if you look at the real application of tensor data, is usually 3D. Okay, so it means three kinds of objects you're looking at is already out of scale. Okay, so and in my lab we are actually working on general tensor models, which can allow any number of type of data to be put together. So we use some low-rank-based parallelized algorithm to explore tensors. Okay, and then the structures I talked about in the network is actually another higher order structure in the tensors. So we are actually exploring those kind of relations among these multi-relations, right? Not just the pairs. Okay, and then we actually, this is what we are working on. And we already have some interesting preliminary results, but I just don't have any that is ready enough to put in a slide. Okay, but this is something actually exciting we're working on now. So, and then just at the end, I want to, so the work actually I talked about by several of my PhD students, like Taehan, Ze, and Wei, and Taehan is now at the Cleveland Clinic, and Wei is at the University of Central Florida. They are faculty members. And my two current PhD students, Rafael and Julio, they are also working on projects relevant to this topic. And then this is also a collaboration with people from Mayo Clinic and U of M, and supported by, previously supported by two NSF grants. Okay, so I think that's the end, and I would like to take questions.
What sort of hardware are you using for your tensor processing?
Okay, so we are just using regular server. We have about 100 core. I think we have 600 gigabyte memory. Yeah, but disk we have 100 terabyte. So yeah, I'm not using GPU because I usually have a high memory requirement. So yeah, my data have to fit into. I need at least 100 gigabytes to run my program. So yeah, usually I'm working on some of the deep learning methods too, and in that case, the GPU is more helpful. But for these tensor methods, we are still using general purpose server. But maybe later, if we have some online methods like similar to deep learning, we can switch to GPUs. Yeah.
Have you, for the tensor calculations, have you found any like third-party tools like TensorFlow, for example? Has that played any role?
Well, because we started work on this before TensorFlow was developed. So a lot of what we are using are our customized program. So this is one library we are using from my collaborator, George Caribis. Maybe some of you took his class in the department. He has a library to the parallelize the matrix tensor multiplication, and one of my models is actually using that parallelized version. This is very, very helpful. Yeah. So.
I guess one more final question. Are you, when you look for relationships and you basically infer a relationship, like you're showing, do you do that in one pass, or do you ever do that recursively and find additional?
Yeah, I think I didn't talk a lot about the algorithmic detail. It is iterative approach where you add new ones, and based on new ones, you try to predict the new ones. But the algorithm has a mathematical framework. So it's not true that we are just keeping adding new ones with the new ones. There's one mathematical framework. There's an objective function we try to maximize. As I mentioned, it is actually, there's a relation among the relations. And that objective function is defined on that. So, but our algorithm just appears to be an iterative algorithm trying to add more association at a time. But it's mathematical driven. So it's not just the intuition. Yeah.
How do you evaluate the association? You said, is it mostly by referring to other literature and saying, yeah, others have reported by other ways, the same associations?
So these general associations are much harder to validate because in the first project, we are very specific. We have ovarian cancer and then with this gene, and then we can look for a cohort, and then we can check for it. So for these general associations, usually it's not necessarily true that we have the data set to do the validation. So it's very hard to do general validations of these methods. And then we also didn't work with any biologists or clinicians to show that there's enough interest. Because if you don't have genetic data, they won't just say, "Oh, you should look at it." They won't believe you. They usually they want to see there are available genomic data. There's sufficient evidence, then they will investigate it. Yeah. So, but I do know people they do this prioritization. So because there are tools they can use, right? So, but they don't have to work with us because the tool is available. You just rank it, and then you can pick the ones you want to study. Yeah.
On a bigger level, like, by learning these associations, are we learning more about the disease, or are we learning more about what we don't know about the disease? Because after a while, if you have so many associations, it seems like there's an...
Both. So that's a very good point, right? Are you just trying to knowledge discovery, or you are trying to make a prediction? Both way. So when I say we are making new predictions, I show the cases where it's designed as a prediction problem. So right, so you have candidate connections, right? Which are the best candidate connections to add? So that's, in a sense, is a prediction. So there's one model, actually, is a good example where we're actually looking at trying to find the module-module relations. So usually people look at the disease gene, whether they're related. And our algorithm is actually analyzes the phenotype network, analyzes the gene network, and then we try to find the group of disease and the group of genes, and then try to find out whether the group of the disease associated with the group of the genes. So, and this is based on the connectivity. So in that way, actually, that's an interesting paper where we show a disease landscape. So, right, we have, so say, 18 different category of disease, and then we have 100 pathways. So then we show these are the most relevant pathways to this category of the disease. So I thought that was, nobody has to do this kind of a global analysis before. We had no way to validate this is true because there are so many diseases and so many genes, right? But it gives you a way of looking at the global picture, right? So if you look at, this is one pathway, and look at the 18 category of the genes, I would say, 3 categories of them might be relevant to this pathway. So yeah, so there are actually, there are discoveries you can make based on the connections too, other than prediction.
What sort of sizes are the matrix and the tensors that you have to multiply, and how many do you have to do for like an iteration?
Okay, so yeah, it's a very high time complexity to do. So it really depends on which problem you look at. So for example, there's one algorithm we developed is to compare multiple networks. So each network in the simulation, each network is 1,000 nodes. And we have 100 of such graphs. So the tensors we are looking for is 1,000 to the 100. So it's huge. But we assume there's a sparse structure in the network. And we use a sparse structure, and we use low rank. So we don't assume there are too many relations we have to look at. But then we are exploring this kind of low rank and sparse structure in the tensors. But you still have to parallelize it. Otherwise, it's still not scalable. Yeah.
Are you guys using supercomputers here on campus to do that?
I'm not. So, well, we have MSI here. They have very good computational resources. But I don't think I can get a hold of, like, so say, 500 giga memories for a couple of days. They won't allow me to do that. You run it for 24 hours and then terminate your process. Right? I kind of have to write a special request so that they can allocate the resource to me. But because I have some good resource in my lab, so we are kind of okay. Yeah. So yeah, but I do want to use a lot of computational resources. But yeah, I also understand because especially memory. So memory is a big limit to some of these methods.
So you're saying like RAM in terms of memory?
Yeah.
Okay. Yeah. So you need over 100 gigabytes.
Yeah, we usually need hundreds of gigabytes. And also, we use when we use these cloud-based platforms, it's not very helpful because there's too much overhead, and it's too slow. Yeah, we're just still using OpenMP, and this is more traditional distributed framework. And then, yeah, so the Spark or these ones actually don't really scale very well. So, yeah, it's probably we don't have enough of these resources to run such a cloud platform. Yeah. Okay. All right. Thank you very much.